Okay, so I just put this assignment available in EduCat. And so it opens today, right after class, and it's like a homework assignment. But it's graded, and I think it's like 15 points, something like that. So it's not super long. Ooh, sorry. Oh, no. So please do it. It's really straightforward, open book, open note, and for points. So this is going to help prepare you for the next exam. These questions are on the CP cycle and the PCO cycle. And I found that having you do this homework assignment before the exam can be super beneficial because it's really kind of forcing you to look at it, think about it a little bit more deeply before you walk in and take the exam. Okay, so make sure to do it. It's not just busy work. It's for points and it's to prepare you for the exam. Okay, so take note of the closing. We generally have been closing quizzes at 11 a.m., not 11 p.m. So make sure that you're clear on it. 11 a.m. is how we're running that for both lecture and lab. Okay. All right, today is going to be split in terms of the lecture because I'm behind in lecture. And tomorrow in lab, you're going to start looking at roots. So I wanted to give you a little bit of information about roots before you walk in there tomorrow. Theoretically, that should have happened, but with being ill and just I'm behind. So what I'm going to do is jump into the roots PowerPoint, which is posted. It's available at EduCat. And I'm going to jump around a little bit because I'm going to give you like this. These are the facts you have to have for lab tomorrow. And then the other remaining part of this hour, I'll go back into our C3, PCO lecture slides. We still have to cover the C4 cycle and the CAM pathway, right? So, but let me start off with root. And so in lab now, you're going to start getting into the different parts of the body of the plant. Whoops. And I like this photograph, somebody laying down on the forest floor and looking straight up. I often think that's what the roots have to imagine, what's above them. And so roots are incredibly important. They're a challenge to study because most roots are buried in soil, but it's a fascinating area of the plant body, and it has so much interaction with its environment in terms of the soil microbes in particular. And so, yes, roots are there to anchor the plant in place, but they do way more than that. And looking at how water uptake is happening through the roots, especially the root hairs, and then also the dissolved minerals that are in the soil water. For both water and minerals, hugely important. And then the roots are constantly, we say roots are leaky. They leak out or exude substances. All kinds of complex molecules will be exuded out of the roots. And we now understand that plants actually have the ability to selectively exude, for lack of a better descriptor, and that allows them to recruit certain types of microbes. So they are not just at the mercy of whatever microbes happen to be there. They can actually alter the ratio and types of microbes around them to create a mutualistic interaction between the plant and the microbes in the soil. So very dynamic, way more dynamic part of the plant body than we ever realized a couple of decades ago. So you tend to see this kind of comparison when you look at overall root system. And I understand this, but there are two extremes. Like on the left is the extreme of a taproot, where it's huge, definite central taproot, very thick, very obvious. And then you have some lateral roots coming off of that. on the right image, you have the fibrous root system where there is no tap root. Instead, all the roots pretty much look the same, and that is very common. Many grasses have that kind of system, but I also want to make you know that there's gradations in between these two extremes. Sometimes it's not so obvious, and so sometimes I see plants that have a thickened taproot, but they also have plenty of more fibrous roots coming off that are pretty thick in diameter too. So it just depends. There's some variation on the design, overall root system design, and we'll get into that a little bit. So if you take a root and you just look at it along its length and look at a longitudinal section, so we're just, it's like you're cutting it right down the length of the root. You can see different zones that are obviously different areas. So at the very bottom of the, or tip of the root, you see the root cap region, and the root cap region is protective. It's very good at protecting that very precious apical meristem region. So this apical meristem is right behind the root cap. Those are a very precious region of cells that are constantly dividing and growing and adding more tissue to the root. And so as the apical meristem is busy doing its cell division and growth, it's adding to the region of elongation, and then the region of maturation is behind that. The region of elongation is just, again, the cells are physically elongating. That's what's going on mainly there. We see the cells are starting to get to their mature size and they're elongating. And then the zone of maturation, that's where we tend to see the cells are very well defined in the role they're going to play for the plant, whether it's an epidermis, vascular tissue, that kind of thing. And so, and then the region of maturation, those root hairs. We tend to only see that region, the maturation region, having root hairs. The more primordial region down low tend not to have the root hairs. So this right picture is what they're showing here, right, is so where you see the protoderm, they're just saying those are cells that will eventually become the epidermis. The procambium, those are the cells that eventually will develop into the vascular. So that's where those, they're just trying to show that really young roots, you know, they start to say protoderm, procambium, but then eventually, especially in the maturation zone, you get, okay, yep, definitely xylem and phloem, definitely parenchyma, definitely epidermis. So yeah, it's just a growth process that they're going through, okay? So you'll be looking at roots and root hairs. And so here you can see, you know, the zone of cell division, the zone of elongation, it's not very big, right? So here's a radish seedling, and if you, so all of this region down here is the root cap, the zone of cell division, the apical meristem essentially, and then the zone of elongation, and then all the rest of the root is the zone of maturation, where you see all those really nice root hairs. So you'll look at those under the microscope and dissecting scope. And so it's, remember, it's the root hairs that do most of the work for absorbing water and minerals. Very, very important, very delicate. So anytime you repot your plants, be nice, but you can be as gentle as you can be, and you're still going to damage a lot of root hairs. It's just impossible not to. Soil is very abrasive, and so repotting your plants is a nice thing to do, but when you get that accomplished, you should water your soil fairly frequently, keep it evenly moist, because it takes them a little bit to regrow their root hairs, and so they're kind of struggling initially. Yes, zone of maturation. Yep. So it's a huge zone of maturation with all those root hairs. So you often see the root area, the main portion of the root. They'll call it the cortex. And just be aware that's just parenchyma cells, a bunch of parenchyma cells. Okay. And so pretty much in this image, almost everything you're looking at is the cortex region. And so, you know, they have this one, pretty much all of this is cortex. And it's just parenchyma cells. It's just cells that they're living, they're busy, doing their day-to-day activities. They also will store excess carbohydrate, for example, and so often you'll see starch granules in there, and so very good. But let me go back. So let's look a little bit more closely. So if you take that root that is in cross section and zoom into the central portion, that's shown on the right. And let me see. So the endodermis is that big red ring, those cells that are in a ring, those are the endodermis. And the endodermis kind of separates out the cortex region of the root to the vascular tissue. All right, it's like a barrier where outside of that dermis you have all those cortex cells. Inside the endodermis you have mostly vascular tissue. The endodermis, I'll show you another picture. The endodermis also has special strip in it, so to speak, in the cell wall that is really waxy. It's made out of suberin wax. The suberin is just a type of wax. And it's laid down in this strip-like formation. I'll show you a picture. And it really, basically what it does is this endodermis with the Casparian strip, the design of it allows for kind of regulating the flow of water and the dissolved minerals into the xylem, right? So it's just keeping everything moving. You know, if you think about the root hairs taking up the water and the minerals, and then the water gets into the cortex region, and then eventually it has to get to the xylem so that the water can start moving through the plant body through the xylem. And so all of that process, the way the endodermis Casparian strip is designed is to keep that moving forward, so to speak, from root hair, cortex, xylem, and just keep that going so that the plant has a nice, easy job of getting water dumped into the xylem, and then it can move. So let me blow that up. So if you take that endodermis, that ring of red cells that you saw in that previous image, this is just looking at a section of that previous image. Right to the inside of the endodermis, there tends to be another circular ring of cells called the pericycle. Okay, the pericycle. So the endodermis is usually stained really nicely red. You'll see that. And then the pericycle is a little harder to see. It's a ring of cells, but it's kind of tricky to find it. But it's another ring of cells right to the inside of the endodermis. And that is an area that can get active and start going through really rapid cell division and growth. And it helps to form, do I have that on here? Is it in the next slide? Where did I put it? Well, let me show you this one. The pericycle can get very, very active. So this region of the pericycle is getting really, really active, and it's growing and forming a lateral root. All right. So if you've ever wondered how lateral roots get made, it actually has to go through the pericycle, goes through cell division and adds more and more cells, and eventually get this elongation of the pericycle to actually see a lateral root coming out from that region. So the pericycle can be very, very important to adding lateral roots to the root system. Okay. Oh, it's in there. It's in the next image down. For some reason that didn't show up on my laptop. So here it is. The lateral root starts to grow from the pericycle. The pericycle is like a meristematic region that kicks into high gear, just grow, 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 and it grows out this nice lateral. Okay. The Casparian strip idea, this is trying to show that. So now again, we're seeing a section of a root in cross section, and you see the root hairs, and then the black line is trying to show water movement pathway. And so they're saying, yep, the root hairs are taking up the water. And the pathway that they've drawn, yeah, if you can look at it a little carefully here, it's roots, water going in between the root cells. You see how it's in between the root cells, it's like hanging out in the cell wall space, laying around in the cell wall space. And that's the most efficient way to get water into the root tissue and then get it to the xylem. The xylem tends to be more into the center part of the root, so it has to go through a pathway of some sort to get to the xylem. And there are like 3 different ways that could happen. The artist here drew the most efficient way, and so this is the quickest way to get the water into the xylem. However, when it reaches the endodermis and that really waxy Casparian strip, it like bumps into the Casparian strip in the cell wall space. It'll bump into that really waxy strip, and it can't go past that. And so that reroutes the water into the cells at that point. And long story short, that action of, oh, I can't go into the cell wall space here, I have to dive into the cell, that, believe it or not, actually helps regulate water flow to keep it moving into the xylem, and not flowing back out of the xylem. You want it to keep going into the xylem. And so that design, very simple design, allows for the water to keep flowing into the xylem and not flowing out. Right? So I won't go into the details on that, but it's a very slick, elegant way of making sure it's kind of a one-way flow of water into the xylem. There's the lateral root idea again that I showed you. Okay, another thing you're going to do in lab is to look at some differences in structural formation of roots. Now with this, we're looking at angiosperms. So we're looking at dicot versus monocot. So anytime you see dicot versus monocot, we're zooming into the flowering plants, the angiosperms. Okay. Now, when you look at an flowering plant, that's one of the first things, if you're trying to figure out what the plant is, you look and say, well, is it a dicot or monocot? And so as we get into more of the anatomy, we'll be making these comparisons. So here you can see whenever you're looking at dicot versus monocot, you want to train your eye to look for the vascular tissue because they design things a little differently. So if you look at the drawing down, so here's a dicot root in cross section. And you can see how the xylem and phloem are smack in the middle of the root. And kind of see how there's a cross configuration of the big xylem cells. So the xylem tend to have that plus configuration. And then the phloem are the more delicate blue-green to my eye. These little patches of blue-green that are in the corners of the cross. That's the phloem. Okay, so you have xylem and phloem clustered dead center of the root. And the xylem's in that plus configuration. So that's what you should be training your eye to if you're trying to figure out, oh yeah, that's a dicot. You're looking for that plus configuration of the xylem, the phloem in the corners of the plus sign, and it tends to be right in the middle of the cross section. So start training your eye for that, and then comparing that over to a monocot root, Monocot, I think about a mono, I think about a ring of tissue, one ring of tissue, a vascular tissue. And that's what you see here. You see that the big cells, solar vessels, there's some tracheas in here too. So we have xylem here in a ring. And then the phloem is, again, kind of that delicate eye blue ring to the outside of the xylem there. So that's the vascular ring of tissue. And so monocots have a single ring like that of vascular tissue toward the center of the cross section here. Whereas the dicot, I think about, the plus has two parts to it. Di means two. And so I just think about the plus having two parts. And so you'll see that you can look at root systems under the microscope and you can immediately, oh yeah, this is a monocot. Oh yeah, this is a dicot, just by looking at how they design their vascular tissue. And then the drawings above it, I'm trying to simplify a little bit what you're looking at. Okay, so you'll look at some different slides under the microscope in lab. So here's an actual dicot root. Again, the xylem pops nicely red in that nice plus configuration. And then the phloem is in the, the phloem is in the four corners. So the phloem is, I can't really see it, I suppose. It's hard to see my laser or my pen. All the purple, by the way, all the parenchyma cells on the outside there of the vascular tissue, that's all ameloplasts. So remember, ameloplasts store starch, and so they stain this root for picking up starch grains. And so they must have applied some iodine because that can stain the root, and starch pops purple. So very, very healthy root that they're looking at here. So yes, go ahead. Yeah. They almost look like they might be sclerides. It's hard to see in the graininess of it. Yeah. Yeah. It does look that way. The endodermis, by the way, is that ring of cells around the vascular tissue. Really hard to see. You just have to kind of squint and look closely for the parasite. So depending on what species you look at in lab, you might see some variations on the theme. So this isn't a solid plus shape, but you can get the gist of it. That yeah, kind of looks like a plus. Right? And so again, this is the dicot root, right? So there are some variations. Not perfect. It depends on the plant and their particular design. But here's another example of the monocot. All right, so whenever we're looking at tissue like this under the microscope, especially in cross section, you're really trying to train your eye to where's the vascular tissue and what is its design. And that can tell you if you're looking at a monocot or dicot, as long as you know some basic, rules of how they do that. Here's a really nice picture showing a monocot root and then the lateral roots coming out of there. Okay, making their way out. So you'll look at some slides like that too. Okay. Questions on the roots? Okay. So in lab tomorrow, please spend some time training your eye, because moving forward now, we're going to keep talking about vascular tissue and what's the design and what's the pattern. And a lot of it is in cross section like this. So make sure that you are getting comfortable with that. I think it breaks through. From what I've noticed on slide preps, it looks like it's kind of obliterated temporarily. It's just the ring of cells immediately inside of the endodermis, but it's really hard to pick out. You know, you have to really look carefully. Okay. So I'm going to come back to this slide set and mention some other things about roots in a little bit here, but I wanted to split my lecture here to go back to the photosynthesis content that we started last week, because we still have some things to say on that. So let me go back to that. Okay, so photosynthesis. All right, so we have worked through the light reaction basics, just the highlights. Then we went into the biochemical reactions that allow for sugar production eventually, but we really have been zooming in on the Calvin cycle, the C3 cycle. So we looked at that last week before the exam. And, you know, this idea that if everything's okay and the plant has the capability to go ahead and open its stomata, then the CO2 starts coming in, and hopefully there's enough RUBP available for the CO2 to react with. And it's the Rubisco enzyme that's really allowing for that to happen. And as long as the Rubisco is seeing plenty of RUBP, plenty of CO2, it's just going to happen spontaneously. You don't have to push any energy towards this reaction. So we've worked through the Calvin cycle. Once that initial step happens, you eventually get that PGA as the first stable product. And then you go through a series of reactions to get your G3P. And to do that, to go from PGA to G3P, you do have to expend some of that ATP that was made during the light reactions and the NADPH made during the light reactions. So it's needed in order to convert the PGA over to G3P. And G3P is really good because it, as you have this Calvin cycle cooking along all day long, you generate enough G3P so some of it can get siphoned off, so to speak, and put into another pathway to allow for sugar synthesis. Sugar. Which sugar do plants really care about? Sucrose. Yeah, sucrose. All right, not glucose. They don't really care about glucose. They want sucrose. All right, and so sucrose is what will get made in another linked reaction pathway. And sucrose is what gets sent through the plant body to different parts to nourish the body, right? So it's sent through as sucrose. Okay, so G3P is really important. You want enough of that building up so that they can send a portion of it off. Then you also see from the G3P section here of the Calvin cycle, you also have to get some of the G3P put into the final steps of the Calvin cycle so you can keep RUBP levels nice and high all day long, because you have to have lots of RUBP for the CO2 to react. Okay, so here you just see arrow, arrow, arrow on step 6. Lots of stuff happening there to take the G3P molecules and convert it to, eventually you do get this RU5P, ribulose 5 phosphate. So it's close, but not quite there. You need one more phosphate. And so you see, again, ATP involved in the light reactions, okay? And then you end up with 2 phosphates. So now you can say, yep, that's RUBP. 5 carbon sugar with 2 phosphates. All right, so we worked through that last week. And then we started looking more carefully at Rubisco. And we said, you know, this enzyme's really kind of funny because it has 2 functionalities. The C on that word stands for carboxylase, which is what's happening here. So what you could say here is RUBP is getting carboxylated, which just means it's reacting with CO2. All right, so carboxylase when Rubisco is doing its carboxylase, it's adding CO2 to RUBP. What does the O stand for? Not carboxylase. What does the O stand for? No, somebody knows. What does the O stand for? Yeah, thank you. Oxygenase. So if we have not enough CO2 coming in for some reason, where CO2 levels start to drop around Rubisco, then Rubisco says, yeah, well, I'll just let oxygen come in. And so here you're seeing on the left what could happen if Rubisco starts to see more oxygen compared to CO2. Oxygen wins, comes into the active site of Rubisco and reacts with RUBP. And you get some funky things going on here. You get a PGA, which is great because that can feed right into the Calvin cycle. What, that 2-carbon phosphoglycolate? That can't go into the Calvin cycle. That's a pain. And so you see that again, arrow, 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 which means a whole lot of stuff happens there to be able to take that phosphoglycolate, and eventually you're going to make another PGA off of that, but it takes a whole PCO cycle to get that to happen. All right, so here again, you're seeing that 2-phosphoglycolate. That's a problem. It can't feed into the Calvin cycle. And so instead it does the PCO cycle, which is this really complicated looking pathway. And so if you look at the top, oh, come on now. There, at the top left, oh, you have the RUBP. They're not showing Rubisco, but it's sitting right where the oxygen essentially is coming in. And then you have the oxygen reacting. Get one phosphoglycerate going to the oxygen. But here's the phosphoglycolate. See that feed in. So complicated because there's 3 organelles to deal with. So these molecules have to get out of one organelle, find the other, get into it, and then start working the reactions in the next organelle like that. So the chloroplast, peroxisome, and mitochondria. And it's in the mitochondria that we have issues here. This is where they say it's a loss of carbon because you see that CO2 there is getting cleaned out of the glycine. And that's considered a loss. It's just respired off. They call it photorespiration because the PCO cycle, they often say it's photorespiration. CO2 is lost in the mitochondria, doesn't make it back to the chloroplast. And so they consider it a loss. If you run the PCO cycle all the way to completion, you can see that you get in this case, they're showing the serine coming into the peroxisome, gets converted to glycerate, which gets over into the chloroplast. Now we're pretty close. So if you just put a phosphate group on that glycerate, that creates feed into the Calvin cycle. But the poor plant had to go through that not once, but twice. It has to run that twice in order to have enough carbon to build the glycerate. Then they have to use extra energy to stick a phosphate on it. So in general, running the PCO cycle is a lot of effort and resources and energy being used. And when they do careful like lab-based studies and make a plant do this, they can see the plant suffers in the sense that it doesn't grow very fast, the yield is low. And so in general, plant physiologists would say you don't want plants running the PCO cycle frequently. And so what happens is, depending on where the plant lives, it could be in an environment where the PCO cycle would run a lot. Like think about anything that would cause CO2 levels to drop in the plant. So what is the plant doing if all of a sudden you see, that plant's CO2 levels inside the leaves is plummeting? What did the plant do to cause CO2 levels to stop? close the stomata. So now think about environments where a plant would be forced to make that decision. Oh boy, I better close my stomata. What kind of environment would that plant perhaps be experiencing? Drought, heat. Okay, so think about areas of the world like the tropical, subtropical area. Yeah, it gets hot through the day, droughty periods, and so closing down the stomata. So if plants are native to hot, dry conditions or droughty, um, periods of drought, heat pretty much consistently is hot, they would run the PCO a lot, and that would not be good. So these plants have evolved a mechanism called the C4 pathway to deal with it. So the C4 pathway is their way to deal with this hot condition, stretches of drought, and basically what it does is it creates a situation where it concentrates CO2 around Rubisco. So that's the goal, or to win the game, essentially. What the plant has to try to create is a situation where Rubisco keeps seeing lots of CO2, very little oxygen. And so they often say it's a CO2 concentrating mechanism. And what they're saying is Rubisco is just seeing lots and lots of CO2. Okay? And if you can maintain that so that Rubisco is just seeing a lot of CO2, then the PCO cycle won't run. And then the plants won the game. Right? So when they designed this through natural selection and adaptation, what they did was they created a spatial separation of certain reactions. They actually put them in different cells of the leaf. And so that's what they're trying to show here, where you have a cross-section of a leaf, and they're showing the parenchyma cells and then the bundle sheath. Remember, these are all parenchyma cells. It's the same thing, just parenchyma cells. And then you have dark green ring of cells there. Those are bundle sheath cells. They surround the vein. Okay, the vein of the leaf, and the vein is the vascular tissue. Okay, so let's look at that. So this arrow is saying we're going to blow up a section where you see a bundle sheath and a mesophyll cell. So this is what I mean by spatial separation. You have some things happening in the mesophyll cell and other things happening in the bundle sheath cell. So here you see the CO2 coming in. So the plant has opened its stomata, the CO2 is coming in, and it makes it into the mesophyll cell. You do not see Rubisco there. Instead, it's a totally different enzyme called PEP carboxylase. Remember, anything with ASE on the end of the word is an enzyme. PEP carboxylase. So this is a totally different enzyme. It's very picky. It only works with CO2. And then the PEP is referring to the idea that the other molecule it works with is PEP, phosphoenolpyruvate, which I think it's spelled out here somewhere. So PEP is a 3-carbon molecule, and then you have CO2. So those two will react, and what you end up with is a 4-carbon oxaloacetate, which then gets converted to malate. Now, it's still 4-carbon. So that's all happening in the mesophyll. Nowhere in there did we say anything about Rubisco, right? PEP carboxylase is very picky for CO2. It does not want oxygen. It can't handle oxygen. So oxygen is not allowed into the active site. Only CO2 and PEP can work into the active site of that PEP carboxylase enzyme. Once malate gets made, it has to move from the mesophyll to the bundle sheath, because its job is to essentially deliver CO2. So I like to think of malate as a vector. A vector is something that moves something from point A to point B in science. Like they often say that the mosquito is a vector for malaria pathogen, So I like to think of malate as the vector for CO2. It's carrying CO2 as part of its structure. It moves physically into the bundle sheath cell and then delivers CO2 right to where rubisco is. You have to imagine rubisco sitting right where that CO2 is, okay, coming in. So rubisco's there like, all right, give me some CO2. I want to see lots of it. And so malate says, yep, I can do that. So malate keeps coming in. As long as this C4 keeps running, you're going to have this delivery of malate and then CO2 given to rubisco. So you see here, the way the artist has drawn it, Calvin cycle is showing up in the bundle sheet. Do you see the Calvin cycle in the mesophyll? No, spatial separation. So the plant is essentially hiding rubisco away from oxygen because what tends to happen in the mesophyll cell, the light reactions are going on in the mesophyll cell. So photosynthesis, the light reactions are going on. But rubisco is over the bundle sheet. The bundle sheet doesn't do light reactions. Right? So rubisco is hidden physically, like separated from where, remember, when the light reactions go on, oxygen is given off. So the plant is it's really well designed to tuck rubisco physically away in the bundle sheath, away from any oxygen getting liberated during the light reaction. And then you have this peroxide acting on the with the CO2 initially, convert it to malate, have malate be the vector to deliver CO2. So it's really kind of a simple little, I like to think of it as like an added on little reaction pathway that's, linked to the Calvin cycle. But it does a wonderful job at minimizing the risk of having to deal with oxygen. It does a great job. So it's not that complicated, and yet it's super efficient. So plants that have the C4 pathway are really good at growing, doing photosynthesis, doing biomass in areas of the world like the tropics and the subtropics where you have hot days consistently and droughty periods. And so they can close down their stomata to like 5% open and just kind of have CO2 trickling in, conserve water, and they can still do great job here with making their sugars. So it's really an elegant solution to this issue of we got to make sure rubisco just sees a ton of CO2. We don't want it to see oxygen. We don't want that silly PCO cycle to keep running. Does it make sense? Yes. I have some data in some future slides here, and actually I just updated that. I need to update that slide set because like the famous example we tend to use is corn or maize does C4. And there's a lot of other plants, but not from a human perspective, not enough. We would like to have more plants do the C4 cycle, the plants we grow for crop production, commercial production, because they're so good at it. They're so good at forming really good biomass. Their environmental conditions that are kind of dicey. And so they're actually trying to make rice a C4 plant. Rice is a C3, and they are, I mean, I just said that's a pretty elegant, simple pathway, but there's a lot of enzymes involved, and so it's tricky. They've been working on it for, I would say, at least a decade now, and they're making progress. Probably within our lifetime, there will be some plant that gets genetically modified to do the C4 when initially it never could do that. But yeah, it's kind of fun to watch their progress. It's the daunting undertaking for sure. This is just another slide. If this helps you study, it's saying the same thing. I think this one might actually be the one from, I can't remember which one's from your book. I tend to teach off this one, but maybe this one's from your book. Okay, so I'm going to go through this quickly. This is part of the PowerPoint, so you don't feel like you have to write all this down. I'd rather you listen to what I'm saying there for you. So here again, this idea that the CO2 is coming in, entering into the mesophyll cells, that PEP carboxylase is there to take it into its active site. It reacts it with PEP, and you get that oxaloacetate. And another way to say oxaloacetate is oxaloacetic acid. So that's where the OAA is coming from, oxaloacetic acid. But by the way, on any quizzes or homework assignments, if I'm asking you to write something, you can just do abbreviations. You don't have to write it all out. And so that enzyme, we say it's very efficient and it doesn't pay attention to oxygen, right? And so that's great. That means that you definitely get PEP and CO2 reacting to give you oxaloacetate, which is what you want, so that you can convert that oxaloacetate to malate or malic acid. Malate or malic acid, same thing. Here it's malic acid figure, and the other one was malate. Anytime you see ATE on the end of a word, that's acid. It's an acid. BSC is bundle sheath cell. So that malate is a vector. It physically moves from the mesophyll to the bundle sheath and is then the CO2 gets cleaved off of it and given to Rubisco. And so Rubisco is like fed that CO2 constantly by the malate coming in. So as long as you do that, you can create a situation where the PCO cycle is virtually non-existent, not running hardly at all. And the downside, though, is that there is some extra input of ATP to get the C4 cycle to run. You have to expend extra ATP compared to the C3 cycle. So it's 2 ATP extra. So if you have a C4 plant like corn and you grow it up here, we're not tropical anymore. So in a situation like this, and there's corn, people grow corn around here. Actually, the C4 plants are at a little bit of a disadvantage because they can't turn the C4 pathway off. They have to run it, which means they have to expend that extra ATP in an area where they really don't need to. Here in this part of the world, we have C3 plants here. This is C3 plant world. And so C4 plants, if you are a grower and you choose to do C4 plants as far north, you'll get a crop, but it probably could do better, more efficient in its true native habitat. This is a cross section of a leaf. I believe this is a maize or corn leaf. And one thing to notice, and when we get into leaf design, I'll point this out again, you can actually tell if a plant is a C4 plant or not by looking at its leaves under the microscope. And the way I can tell right away, I go, I think this is a C4 because the bundle sheet cells, you know, back there, they're 2 feet full of chloroplasts. That's a dead giveaway that you're looking at a C4 because they really beef up their bundle sheet. All plants have a bundle sheath around the vein, but these are like really big and lots of chloroplasts. And then you tend to see another thing too is that the artist draws the bundle sheet quite as big as it should be. But one thing they did nicely is they're showing the parenchyma cell with kind of hugged up to the bundle sheath, which makes sense if you have to get malate into the bundle sheath and then, you know, finish the cycle. It makes sense if they're physically well next to each other. There's plasmodesmata where they can just keep circling. Remember, they got to keep going. You have malate coming into the bundle sheath and coming back. So it makes sense that they're hugged up to each other. So that's another thing you look for too, is that you have good connections between the bundle sheath and the surrounding parenchyma cells. C4 plants, sugar cane, which again is a big commercial crop. is C4, corn is the big picture there. In the lower right, that's sorghum. Sorghum is right up the river. I'll update the slides on the C4 idea and repost these. But so then, but today's Wednesday, so Friday we'll get into the last pathway, the CAM pathway, and luckily it's very similar to the C4 pathway, so that'll be pretty straightforward. Yeah, so I'll talk more about the